So now we're entering into a technological phase of our meeting. We're going to have a WebEx presentation from Professor Mildred Cho, who's out at Stanford, where she's a professor of medical genetics and pediatrics, and also the associate director of the Stanford Center for Biomedical Ethics. And I'm just tap dancing here to see if we can pick her up. I don't know where Mildred is today on this WebEx. She could be at the office or out in her house, I suspect, to do this. And I think she's been listening in on us.
Hi, Art. I'm here. Can you hear me?
Here you are. How are you?
Great.
How's the dog?
Very good.
And we see your slides, and I think what we're going to do is ask you to say next slide and so on when you're ready. I'm just going to stand here 1 second to make sure this goes, but you can start, Mildred.
Okay, thank you. Thank you, Art, and to the committee for inviting me and allowing me to participate from the wilds of Utah remotely. So hopefully this will work. So I'm going to try to go through my presentation really quickly so that we have some time for discussion. So I was asked to talk about the ethics of germline gene editing in non-human primates. And I think one thing that distinguishes my presentation from some of the others is this is still, the use of gene editing in non-human primates is still in its infancy, especially in terms of the germline uses. So next slide, please. So we really can talk a little bit about some of the issues that were raised some years ago, such as back in 2001 when a transgenic rhesus monkey was produced by transgenic methods using a replication defective retroviral vector. And one transgenic animal was born. 3 males were born, and one was transgenic in all accessible tissues. But germline transmission or tissue expression was not confirmed. Next slide. But then in 2009, a lentiviral vector was used to create transgenic marmosets with, again, the green fluorescent protein shown here. And the neonate expressed the transgene in skin, but not in placenta or hair, so not in all tissues, but there was germline transmission. Next slide. In 2013, the group that produced the rhesus monkey that I showed you on the 2 slides ago was produced a rhesus macaque using a lentiviral vector carrying instead of just the green fluorescent protein, exon 1 of the human Huntington gene and the green fluorescent protein. And germline transmission was confirmed at least by production of embryos and embryonic stem cells from the sperm of the transgenic monkey. Next slide. So what's been happening with respect to gene editing in monkeys? There's been a lot of activity in recent years along these lines using both CRISPR and TALEN's approaches. Next slide. So in February of 2014, researchers targeted multiple genes at the same time, including the NR0B1 transcriptional regulator that's associated with congenital adrenal hypoplasia, among other things, the PPAR gamma gene, which is a nuclear regulator of fatty acid metabolism and glucose metabolism, and RAG1, which is involved in activation of immunoglobulin VDJ recombination. So this was a multiplex approach that was used to try to modify 3 genes simultaneously. They were successful in deriving animals that had modifications in the PPAR and the RAG1 genes, but not in the NR0B1 gene. And no off-target mutations were detected, at least at 84 potential sites that were analyzed here. So this was interesting in that this gene editing technology does allow some new approaches to be tested using multiplex models for human disease. However, it wasn't really clear or described by the researchers why these 3 genes in particular were chosen for these experiments, although they are clearly associated with human diseases. It wasn't really clear why these 3 in combination were chosen. Next slide. And in these animals later, there was confirmation of expression of PPAR gamma and RAG1 in the testis tissue of 2 male embryos and ovary tissue in at least 1 of the 2 female embryos that were derived, but no direct evidence for germline transmission in live animals was obtained yet at that stage. Next slide. And so, as Art mentioned earlier, there have been a number of human diseases that are attempted to be modeled in research animals in non-human primates, including Rett syndrome. So there was a paper published in 2014 using the TALEN system to mediate mutagenesis of the MECP2 gene associated with Rett syndrome, which is an X-linked condition in which loss of function mutations generally lead to not only intellectual disability and as a neurodevelopmental disorder, but also loss of the use of hands and problems with walking as well as seizures. So it is a very complex condition in addition to the cognitive functions that are affected by the MECP2 gene. And so in this study, there were fetal tissues analyzed that included testis tissues that did appear to carry the MECP2 mutations. And the MECP2 mutagenesis was also detected in placental and umbilical cord, as well as skin tissue being more accessible, and no off-target loci were detected here. Next slide, and maybe skip on to the next slide. So just as examples of other genes that are of interest for development of human disease models in non-human primates, P53 is one gene that has been analyzed in cynomolgus monkeys, and in this particular case that was published in February of 2015, animals with biallelic mutations of the P53 tumor suppressor gene were generated using the CRISPR-Cas model. And so this allows for null mutations to be developed using this model, which is potentially useful for more precise modeling of human diseases. Next slide. So in April of 2015, the CRISPR-Cas system was also used to induce mutations in rhesus monkeys in the dystrophin gene associated with Duchenne muscular dystrophy. And in these models, both male and female monkeys were generated with pronounced muscle degeneration at birth, which is different from the pathology that's displayed in humans, which has a bit later onset, in contrast to mice who display milder symptoms and milder pathology. So the monkey model could be a better model of human disease. However, the Cas9 and TALENs-mediated gene editing appears to produce mosaic mutations in both humans and non-human primates. So the utility as a disease model may be limited, even though Duchenne muscular dystrophy, being an X-linked condition, is also mosaic in females, but not in males. Next slide. So on to the ethical considerations. I think Art already described these very well at the beginning, so I'm sort of bringing us back to some of the issues that he's already raised. And also, in the spirit of what Allison had talked about in her presentation, I think it's important for the purposes of this presentation and this conference to think about what is unique about, first of all, gene editing as a technique versus other transgenic methods, and what is unique about germline versus somatic genetic modifications. Germline gene editing has the potential to lead to better disease models, as Art mentioned, especially for the study of loss of function mutations and genetic interactions, and the multiplex genetic manipulations that I described earlier. However, this technology also has the potential to produce more serious disease in animals, as Art mentioned. And I think he described this catch-22 phenomenon very well already. The germline gene editing also has the potential to facilitate breeding of animals for human disease modeling, so potentially beneficial in that way. Next slide. But again, I think this slide captures what Art already described as the ethical catch-22, which is that we would like to use non-human primate models of human disease precisely because non-human primates are closer to humans in many ways. And we are also interested in human lineage-specific genes. That is, what are the genes that are specific to humans that make humans behave and act in the way we do, which might be different from other primates, and be interested in using those genes for gene editing purposes to test them individually in non-human primates. Next slide. So if we take the strictly utilitarian view, we would be weighing the likelihood of benefit with risks. And it's important to think about what kinds of risks we're talking about. I'm going to focus right now on the risks to non-human primates, but some of the other risks that we also need to think about are the risks to potential other animals in the environment, and the risks to humans. So, for example, the risks of transmission of diseases to humans through vectors that infect humans, such as adenoviruses, must also be considered if they are developed for use in non-human primates, but might also affect and infect humans. Next slide. So I think it's useful to think about a report that the Institute of Medicine, along with the NRC, developed in 2011, addressing the issue of the use of chimpanzees in biomedical and behavioral research, where they, I think, did a really good job of coming up with a framework for assessing the scientific necessity to guide the future use of animals, and think about how we might use that framework for other non-human primates. And the biomedical research criteria that they proposed in this report was that no other suitable models are available, that research cannot be performed ethically on human subjects, and that foregoing chimpanzee research would slow or prevent advancements to prevent or treat life-threatening or debilitating conditions. So these are pretty strict criteria. And as of this year, the NIH has now, as you probably know, has ended its support for chimpanzee research. Next slide. In the report, they did the committee did talk about biomedical research using stored samples as being exempt from these considerations because of the lower risk of using, for example, blood samples or tissue samples, but stressed that animals must be maintained either in ethologically appropriate physical and social environments or in natural habitats. And that is assuming that the potential for non-human primates to suffer from induced diseases is likely higher than other animals because of their complex social, cognitive, sensory, and motor functions, and the likelihood of benefits from gene editing and germline gene editing must be carefully and realistically assessed and not assumed. Next slide. So in the report, they did talk about the utilitarian justifications for for continued research in animals, that is, animals being subjected to risks for the benefits of humans, requiring justification of benefits outweighing the harms, which we've talked about. Next slide. But they also talk about the purely utilitarian justifications needing to be tempered through policy requirements for humane treatment and appropriate species uses and minimal number of animals, and also the moral costs that Art has already mentioned. Next slide. So the conclusions in this report were ultimately that while chimpanzees have been a valuable animal model in past research, most current uses of chimpanzees for biomedical research is unnecessary based on the criteria established by the committee. And they did list a couple of specific examples, which are very, very narrow uses, and as well mentioned that new or emerging or re-emerging diseases may require future uses of the chimpanzee. Next slide. But I think the value of this report was that it did discuss the issue of assessing likelihood of benefit. I think when we talk about weighing risks and benefits, we tend to focus on the risks. But I think one of the things that could be improved in the thinking about uses of non-human primates is assessing the likelihood of benefit, and in this case, the benefit from gene editing technologies. And so there's been some attention, but perhaps not enough attention to how relevant non-human primate models are to human diseases. People have pointed out that non-human primate research has not been particularly good at predicting human responses to viral vaccines, possibly because immune systems greatly vary between species, that there could be human specificity of some neuropsychiatric conditions, and that there are limited cognitive behavioral tests for non-human primates, such as marmosets, and some of these primates have small head sizes, which makes imaging and other kinds of diagnostics difficult. And in addition, improving cross-species predictability of behavioral tests is necessary, for example, to distinguish state from trait anxiety in tests of anxiety. Next slide. More recently, people have pointed out that there needs to be more attention to the species-specific genomic variation patterns in non-human primates as compared to humans, because genetic variation in non-human primates is much more extensive than variation in humans, but less well understood. There are very few trans-species polymorphisms in humans and chimpanzees, for example, and implications of variation in genes that are very important for developing models for drugs, such as cytochrome P450 genes, and their effects on drug metabolism in non-human primates is not well known. Next slide. So people have been pointing out lately the need to examine study designs to improve the likelihood of benefit of animal studies and their relevance to human models, because I think there is some consensus that animal studies of drugs or biological treatments are much more successful than human studies in general, and we still need to do more work on understanding why that is the case. Next slide. So, for example, it's been shown that animal studies, similar to human studies, that score higher on study quality checklists produce substantially lower estimates of effect size than lower scoring studies. In this diagram, the higher scoring studies are on the right, and the lower scoring studies are on the left, and you can see that the effect sizes are systematically lower with the higher study quality. Next slide. And as another example, non-randomized studies, shown here on the right side and the top, have higher estimates of efficacy than randomized studies. As well, studies without blinded outcome assessments have higher estimates of efficacy than blinded studies. And these diagrams show examples of studies, animal studies of acute ischemic stroke. And these are estimates of infarct volume in which the estimates of volume were 27% higher than randomized studies in the non-randomized studies. And studies without blinded outcomes had 19% higher estimates than blinded studies. So these are similar findings to those that we've seen in human studies. So I think this suggests that improvements in study design could make the animal models more relevant. Next slide. So this is my final slide. So I think that germline gene editing of non-human primates raises similar ethical issues as those raised by other transgenic methods. It's more sort of a matter of degree than quality of the arguments. But gene editing does have the potential to broaden the array of non-human primate models of human diseases, which carries benefits with it, but also potential risks to the non-human primates because these could be more debilitating phenotypes. And so induction of serious disease can only be justified by careful and realistic assessments of benefits likely to accrue from non-human primate models and use of rigorous study designs, similar to what we would say for human studies. And I think that the transparency that many of the previous speakers have talked about to enhance public trust, both on the part of researchers using non-human primates as well as the IACUCs, would be helpful in this regard. So that's it.